Hello guys, welcome back to my YouTube channel Excellence Hub. Today I'm going to continue the topic artificial chromosomes. Today I'm going to discuss about yeast artificial chromosome, that is YAC, and its library construction.
Yeast artificial chromosome. These are also genetically engineered chromosomes derived from the DNA of the yeast. It is also a human engineered DNA molecule used to clone DNA sequences in yeast cells. They are the products of a recombinant DNA cloning methodology to isolate and propagate very large fragments of DNA in yeast host. What does this mean? Yeast artificial chromosome. These are also the vector that help to produce recombinant DNA molecule by inserting large foreign DNA fragment into it. And here host cell is yeast. By inserting large fragment of DNA, the inserted sequences can be cloned and physically mapped using a process called chromosome forking. Amount of large foreign DNA, large foreign gene of interest that we can ligate into the YAC vector system is about 200 to 500 kb. As much as it can uptake 1 megabase pair that can be cloned into YAC vector system. Various components of BAC, centromere. It ensures chromosome partitioning between two daughter cells and a selective marker genes. ARS, autosomal replicative sequence. Yeast telomere. It also has selectable markers such as URA3 and TREP1. And bacterial selectable marker is also present in it. This is the general overview of the YAC vector system. This is one of the restriction site. This is the another restriction site. These are centromere. This is autosomal replicative sequence. Various selectable markers. Yeast selectable marker. One is TRP1. Another is URA3. And this is bacterial selectable marker. This is how the YAC is being constructed. This is the two restriction site. One is of EQR1, another is of BAMH1. EQR1 will going to digest here and cut from this, and BAMH1 will going to come here and digest this and make a cut in here. So now when both the restriction enzymes will going to work, this YAC vector, the circular YAC vector, is going to be produced two linear fragments. The two telomere sites that are present at both the sides of the restriction site 2 BAMH1 are going to be at the both the ends. One is here and another is here. And in between, sticky ends are being produced. And these sticky ends help that large foreign insert can be inserted here easily. This is how the construction of library can be done. We have studied that the two restriction enzymes are going to cleave the two sides and produce two different fragments having at both the ends have sticky ends that help to integrate this large DNA fragment into it using ligase enzyme. This is the process that we have studied. YAC vector. YAC vector is initially propagated as circular plasmid inside bacterial host utilizing bacterial ori sequence. Circular plasmid, YAC circular plasmid is cut at specific site. Two restriction sites are there using restriction enzymes. When both the restriction enzymes work, it's going to produce linear chromosome that has two telomere linear sites at both the terminals. Now this linear chromosome is again digested at specific site with two arms with different selection marker. Now it's going to produce sticky end that help to insert genomic DNA to ligate into it using DNA ligase enzyme. The recombinant vector that we are going to obtain is being transformed into yeast cells, and now we are going to screen using selection marker to obtain recombinant cloning to differentiate between recombinant and recombinant. Now, various advantages of using YAC vector system. It provides largest insert capacity of any cloning system, and various YAC vector system that are produced YACs, YAPs are more advantageous than BAC. They can be used to express eukaryotic proteins that require post-translational modification. Various disadvantages of using YAC vector system. These are very large DNA molecules and very fragile and prone to breakage, leading to problem of rearrangement. It has high rate of loss of the entire YAC during mitotic growth. It is difficult to separate YAC from the other host chromosomes because of their similar size. Separation requires sophisticated pulse field gel electrophoresis, PFGE, pulse field gel electrophoresis. Yield of DNA is not high when the YAC is isolated from yeast cells. Clones that we obtain are unstable with their foreign DNA inserts often being deleted from it. Then various applications of YAC library. Applications of YACs include generating whole DNA libraries of the genome of higher organism. It also helps to identify essential mammalian chromosomal sequences necessary for the future construction of specialized mammalian artificial chromosomes, MACs. By being able to insert large fragments of DNA, YACs can be utilized to clone and assemble the entire genome of an organism. With the insertion of YAC into yeast cell, they can be propagated as linear artificial chromosome. Cloning the inserted region of DNA in the process. With this completed, two processes can be used to obtain a sequenced genome or region of interest. One is physical mapping, another is chromosome walking. We have studied about BAC system and YAC system. This is the table of comparison between two vector systems. First, on the basis of host cell. In YAC, its host cell is Saccharomyces cerevisiae. In BAC, host cell is Escherichia coli. On the basis of type of DNA molecule in them, in YAC, DNA molecule is linear, and in BAC, it is circular plasmid. Insert size, YAC can take up to 1 to 2 megabase pair of foreign DNA fragment, and BAC can take up to only 300 kb. Protocols for handling and isolation of DNA. YAC, it has difficult protocols. BAC have easy protocols. DNA yield in YAC, it is low. In BAC, it is medium. Resistance to shearing, it's low in YAC and it's high in BAC. Direct sequencing possible. In YAC, it is not possible. In BAC, it is possible. Selection markers in host cell. In YAC, complementation of auxotropic mutant, resistance to drug. In BAC, resistance to antibiotics such as kanamycin or chloramfene. Insert chimerism, it's high in YAC and it's low in BAC. Insert rearrangement, it is possible in YAC, it is not very rarely, it's not possible in BAC. Modification capabilities in YAC, it's plenty, reproducible protocols, and and in back it has few protocols in evolution. Protocols for mutagenesis, it's easy in YAC, it's difficult in back.
Thank you guys. Do subscribe the channel, hit the like button and share the video. Here the artificial chromosome library is being complete. Thank you.